Hello and welcome to the presentation of our chapter on the genetics of cerebral cavernous malformations. We will begin with a brief overview of early work, then spend some time discussing the genetics of CCM, briefly touching upon the function of causative genes and their encoded proteins. We will take a look at the cellular and molecular pathways to which CCM protein function is integrated, and discuss our current understanding of disease pathogenesis and approaches in preclinical models aiming to identify therapeutics. The earliest known case of a cerebrovascular lesion, a berry-like growth within the frontal lobe, was described in 1854 by von Luschka. More than 70 years later, Cushing and Dandy independently reported extensive patient series with cerebrovascular malformations, which were subsequently classified into 4 distinct histopathological subtypes. The focus of this chapter is on cavernous malformations, which are also known as cavernous angiomas or cavernomas, or as cerebral cavernous malformations, for brevity, CCMs. CCMs are common lesions in the CNS and rarely the retina. They have an estimated population prevalence between 0.3 and 0.9 per 100 individuals. They are mostly clinically silent, with only about a third becoming symptomatic, and depending on location and size, CCMs can cause a broad range of symptoms, including headache, seizures, and focal neurological deficits. A fraction of patients with CCM experience catastrophic, potentially fatal cerebral hemorrhages. Both sporadic and familial forms of CCM exist. CCMs are collections of enlarged, densely packed vascular sinusoids with abnormal structure. They are lined with a single layer of endothelial cells, they lack vessel wall elements, and they're embedded in a collagen matrix without intervening normal parenchyma. CCMs can be found in all brain regions and are usually 1 to 5 centimeters in size. Due to their primitive vessel architecture and low pressure blood flow, CCMs tend to leak, causing microhemorrhages and subsequent hemosiderin deposition. Treatment options include observation of asymptomatic lesions, anti-epileptic medication, surgical excision of accessible lesions in patients with symptomatic hemorrhage or intractable seizures, and radiosurgery. However, pharmacological therapies that improve outcome are not available. Sporadic CCMs are more common and usually solitary, whereas familial CCM is characterized by multiple lesions. The hereditary nature of CCM was recognized as early as 1928, when Koofs described a father and daughter presenting with cerebral, retinal, and cutaneous cavernous malformations. Familial CCM is characterized by autosomal dominant inheritance and high but incomplete penetrance in the setting of strong family history. Numerous families have been reported around the world. Genetic linkage analysis performed in the 1990s mapped 3 CCM loci, and subsequent efforts identified the causative genes: CCM1 or CRIT1 on chromosome 7q, CCM2 on 7p, and CCM3 or PDCD10 on 3q. Loss of function mutations in these 3 genes account for 98% of familial CCM disease. Although they are generally thought to lead to clinically and radiologically similar disease, familial CCM associated with CCM3 mutations is characterized by exceptionally aggressive clinical phenotypes, greater lesion burden, and more frequent hemorrhages earlier in life, leading to the designation of the CCM3 syndrome. CCM1 or CRIT1 is the largest of the three CCM proteins. It appears to lack catalytic activity and contains several domains implicated in protein-protein interactions. CCM2, similar to CCM1, has no enzymatic activity, and it binds to CCM1 and to CCM3 at the same time. CCM3 is the smallest of the three proteins, and it has a variety of well-documented interacting proteins. A series of studies from many labs demonstrated that the three CCM proteins are essential for vascular development, and that CCM1, 2, or 3 deficiency, specifically in endothelial cells, leads to brain vascular malformations in mice. Importantly, however, vascular lesions also form when CCM3 is inactivated in other cell types as well, for example, radial glia progenitors, astrocytes, or pericytes. Our understanding of the cellular and molecular mechanisms underlying function and malfunction of the CCM proteins is advancing, but remains far from complete. CCM1, CCM2, and CCM3 can exist in a dynamic trimeric complex with CCM2, which is a scaffolding protein, acting as the hub and interacting directly both with CCM1 and CCM3. However, each of the three CCM proteins also interacts with a variety of other signaling, cytoskeletal, and adaptor proteins. There is now substantial evidence that loss of CCM genes or CCM protein disruption leads to a common biochemical defect, activation of ROA signaling and its effectors ROC1 and 2. It is also true that loss of CCM genes leads to hyperactive MEK-K3 signaling and its downstream targets KLF2 and KLF4. Now, a few words on the mechanisms of CCM pathogenesis. Familial CCM is an autosomal dominant disease. Knudson's two-hit mechanism, originally proposed for retinoblastoma, has been employed to explain CCM pathogenesis. According to this model, the first mutational event is inherited in the form of a germline mutation, whereas a second mutation is acquired in a subset of the somatic cells, which can no longer synthesize functionally intact proteins, rapidly leading to disease. Support for this model comes from observations in patients, including one with post-zygotic mosaicism. The caverns, in this case, are lined with a mixture of endothelial cells that are either heterozygous, carrying a germline mutation, or compound heterozygous, carrying one germline and one somatic mutation. Support also comes from analysis in mouse models, where clonal expansion of a heterozygous endothelial cell that acquires a second somatic mutation proliferates to initiate lesion formation, while also recruiting neighboring wild-type cells contributing to lesion growth. In the sporadic form, both mutations occur in somatic cells, and next-generation sequencing has identified mutations in the three CCM genes in sporadic CCMs. However, very recent work has suggested that a significant fraction of sporadic CCMs harbor somatic activating mutations in known oncogenes. Advances in our understanding of CCM biology are beginning to inform the rational exploration of possible therapeutic approaches, ranging from targeting of signaling pathways that are dysregulated in CCM disease to unbiased drug screens. For example, Pharmacological inhibition of ROA-ROCK signaling continues to be explored as potential therapeutic strategy for symptomatic CCM patients. The upregulation of beta-catenin transcriptional activity following CCM3 loss inspired the evaluation of two anti-inflammatory drugs, sulindac sulfide and sulindac sulfone. The hyperactivation of MEK-K3 signaling in CCM, on the other hand, inspired a search for approved small kinase inhibitors that identified ponatinib, a drug currently used for acute myeloid leukemia. Propranolol, a beta blocker used for the treatment of proliferating infantile hemangioma, has been proposed for the management of patients with symptomatic CCM. On the other hand, unbiased screens of approved drugs that can be repurposed have also been carried out. These identified cholecalciferol and tempol, respectively vitamin D3 and a scavenger of superoxide, fluvastatin and zoledronic acid combination was found to be effective in chronic and acute models of CCM3. Whereas indirubin 3 monoamine, an FDA-approved drug which is derived from traditional Chinese medicine and is used to treat leukemia and other chronic diseases, was identified using a multi-organism-based approach. Considered together, targeted as well as unbiased approaches to identifying CCM therapeutics are slowly pointing to candidate drugs for further evaluation. What should be noted, however, is that these candidates have yet to be tested across all mouse models of CCM, for example, harboring mutations in the 3 CCM genes, in different cell types, and in the context of acute or chronic disease. And thus, whether they are broadly effective, even in model organisms, remains an open question. While many questions remain unanswered, CCM is a prime example of how investigation of a familial form of a disease may help reveal its pathophysiology through genetic studies followed by analysis of the cellular and molecular mechanisms underlying disease pathogenesis. This understanding will provide important insight into both familial and sporadic forms of disease by dissecting the involved molecules and pathways, which ultimately will be the substrate for novel rational therapeutic approaches and possibly disease prevention. Currently, there are 2 active clinical trials, a phase 1/2a single-site trial, which will evaluate atorvastatin versus placebo in stabilizing CCM after symptomatic hemorrhage, and a small-scale exploratory trial pursuing propranolol. I would also like to take a minute to bring to your attention the Angioma Alliance, whose mission is to inform, support, and mobilize those affected by cavernous angioma and drive research for better treatments and a cure. In addition to Angioma Alliance, many other alliances exist throughout the world. Please visit their web pages for more information. Finally, a few key points to remember. CCMs are low-flow cerebrovascular malformations with high prevalence. Sporadic CCMs are more common and are usually solitary. Familial CCM is characterized by multiple lesions, which are due to loss of function mutations in CCM1, CCM2, or CCM3. The 3-CCM proteins are critical for vascular development, and their deficiency leads to activation of RhoA and MECK-K3 signaling. Emerging therapeutic options are still being evaluated in preclinical models and early-stage clinical trials. We would like to thank all those whose work is cited in the chapter, the patients and their families, and funding for CCM-related work. Thank you for listening to our chapter.